Okay, so here's my labeled picture. So here you have a neuromuscular junction or synapse. So the term neuromuscular junction is really an anatomical term because it's telling us, the union of what kinds of cells. So you have a union between the neuron and the muscle. So neuromuscular junction. A synapse is more of a functional term because it's telling us what a synapse that's the functional union or functional union between a neuron and its effector. So in this case, the effector is a muscle, right? So it's a neuromuscular junction, or what's taking place here is called a synapse. That synapse is going to ultimately lead to contraction. So labeling some items we have here, in yellow, the motor neuron, which I've labeled up here. This portion of the motor neuron is called the axon. It's along the axon that the action potential is delivered, and that's initiated in the brain. So the brain says, I need this muscle to contract. So the brain will send an action potential along the axon of this neuron. It will travel towards the axon terminus, located here at the end. It's within the terminus that we find the synaptic vesicles. So these gray round circles are synaptic vesicles. And they contain, as long as this is a skeletal muscle, they will contain acetylcholine. Acetylcholine can be abbreviated capital A, lowercase c, h, ACH for acetylcholine. So that's a neurotransmitter housed within those synaptic vesicles. The synaptic vesicles will release acetylcholine into the synaptic cleft, which I neglected to label, but that's that little space right there, the synaptic cleft. ACH will diffuse across the cleft and bind to ACH receptors located here. They're embedded in the sarcolemma. Now, I did go ahead and label sarcolemma right here for you. Sarcolemma is what I've colored in brown. But the portion of the sarcolemma right here that contains the ACH receptors, this is referred to as the motor end plate. So the motor end plate, let me just add that label really quickly. I'm going to spin it around because I'm upside down. Motor end plate. Okay, there we go. So that's the region of the sarcolemma that contains the ACH receptors. Okay. When acetylcholine binds to those receptors at the motor end plate, that will initiate another action potential, this time along the sarcolemma. There are extensions of the sarcolemma that extend down into the sarcoplasm called T-tubules, or transverse tubule, or for short, just T-tubule. So the T-tubule will transport that action potential down into the interior of the muscle cell. They travel right alongside organelles called the sarcoplasmic reticulum. So I've labeled that here, sarcoplasmic reticulum, or you can abbreviate SR. I've kind of colored the boundary here in dark red. The sarcoplasmic reticulum is a specialized organelle found in muscle cells that stores and releases calcium. That's its big job, to store and release calcium. So the calcium is colored in blue, so they're the blue dots. The calcium will be released from the SR in response to the action potential traveling down the T-tubule. So when the action potential moves down the T-tubule, that will trigger the release of calcium ions from the sarcoplasmic reticulum. The calcium ions will move to the sarcomeres and bind to troponin. So here's our sarcomere. This is our portion of it. Here's our actin, the myosin in blue. These are the myosin heads or cross bridges. There's our ATP. So when calcium binds to troponin, it's going to allow for the binding of the myosin heads into the actin active sites. I'll discuss that detail in lecture. I just want to go ahead, I wrote on here that muscle contraction requires calcium, all right, and it will require ATP. So we'll talk about ATP. ATP actually re-energizes these myosin heads, so we will talk about that phenomenon in lecture as well. But if you can get that anatomy down, that will be very helpful when we start to talk about the entire contraction process. Okay, there we go. That should be the end of your document.